Hello, Interstellar everyone. Today we are going to review our biology sample paper for year 2020-2021. Right. So this will be a quick review of the sample paper. Right. What are the pattern changes that have been made for this particular academic year? Right. For your board examination, what kind of question paper that you can expect? So you will know from this particular review. Okay. So by looking at the sample question paper 2020-21, we can see over here in the general instructions that all the questions given in the question paper, they are compulsory. Right, there are a total of 33 questions divided into four sections: section A, B, C, and D. Right, and the question paper is out of 70 marks. Right, so it carries a total of 70 marks. There are 33 questions divided in four sections: A, B, and C, A, B, C, and D. Right. Section number A has got 14 questions which carries 1 marks each, and there are two case-based questions. And there is a section B which has 9 questions of 2 marks each. Section C has got 5 questions of 3 marks each, and the last section D has got 3 questions of 5 marks each. There is no overall choice, but there is internal choice which have been provided in some of the questions. Right. And in such case, a student has to attempt only one of the alternatives in such questions. Okay. And you have to draw neat and properly labeled diagram. Like I always tell you during the class time, you have to practice drawing diagram. And in answer sheet, you have to write neat and properly labeled diagrams. I told you how to draw the diagrams and how to label them. Right. If possible, if there are less labels, try to label them on one side of the diagram so that it looks neat. Okay, so these are the general instructions. Now, if you look at the previous year's board examination sample paper, you will see the difference, right? For example, in section number A, earlier there used to be only 5 questions which carried 1 marks. Now we have got 14 questions which carries 1 mark, and then over that there are 2 more questions which carries 4 marks each, but those questions are of 1 marks each. So we will see later on. So 1 marks question, there will be about 22 questions. Right? I think it is 22, right? If you add 8 over here, there will be 22 questions which carries 1 marks each. Right? So that is the change. That is a major change. And in section B, earlier there used to be 7 questions which carries 2 marks each. Now there are 9 questions which carries 2 marks each. So there is a little bit increase. 2 questions have been increased over here. The major changes is in section A and section C. Now section C, earlier there used to be 12 questions of 3 marks each. That's a lot actually. Now it has been reduced to only 5 questions of 3 marks each. And the last section D is same. There are 3 questions which carries 5 marks each. And other instructions are same. You have to draw diagram, neat and label diagram. Now I told you there are 33 questions, right, which carry 70 marks in total. Section number there are 4 sections: section A, B, C, and D. Right. In section A, right, from question number 1 to 16, it is included in section A. Question number 1 to 16, they are included in section A, and they all carry 1 marks, which means total of 22 marks. Now, apart from number of questions with 1 marks, what else has changed? Right, that is the question. Section A, what else has changed apart from the increase in number of questions? Earlier there used to be only 5 questions which carries 1 marks, right? Now there are many questions, 16 questions which carries about 22 marks. So the thing that has changed is types of questions. That has changed over here, right? And 2 major changes are assertion reason type of questions are included in your question paper. Right? We will see what do we mean by assertion reason type of questions. And then there is case-based questions that we will also discuss in this video. So the first one is assertion reason type of question. But before we discuss that, the first 10 questions, they are just like normal 1 mark question, which you already know how to answer, right? You don't have to write more than 1 or 2 sentences for such type of questions. If you know the keyword, you have to include that keyword. Okay, so question number from question number 1 to 10, it all carries 1 marks each. Right? These are short answer type of questions. Right? Very short answer type of questions. You can either write 1 word, one sentence or two sentences at the max. Don't spend too much time on each question. Okay, so that is the case. And then later on, from question number 11 onwards, right, the changes have been included, like inclusion of assertion reason type of questions, right? So what do we mean by assertion reason type of questions? So if you are familiar with this type of question, well and good. If you are not familiar, try to read over here. Assertion reason type of question, which means there are two statements given in such type of question. In such type of question, there are two statements given. One is assertion, another one is reason, right? So if we look at question number 12 in your sample paper, there are two sentences given over here. First one is the assertion. Assertion, it says E. coli, Escherichia coli, which which have PBR322 vector or the plasmid DNA with the DNA inserted at BAMH1 site cannot grow in a medium containing tetracycline antibiotics. So that is the assertion. And the reason provided for this particular statement is the recognition site of BAMH1, which is a restriction enzyme. It is present in tetracycline R resistance region of PBR322. Right? So there are two sentences you can clearly see over here. Now, after reading these two sentences, you have to make a selection from A, B, C, and D. So it is like a MCQ, but it's a different kind of MCQ, right? Over here, option number A, it says both assertion and reason are true. Right? Both assertion as well as reason are true, and the reason is the correct explanation of the assertion. So you have to check it out whether it is true or not. And then option B says both assertion and reason are true, but the reason is not the correct explanation of the assertion. Right? Whether reason given over here, whether it supports the assertion or not. So this is the thing that you have to check. And in option C, assertion is true, but the reason is false. So the statement assertion can be true, but the reason can be false. And the option D says both assertion and reason are false. So now this checks your knowledge and how you apply that particular knowledge. Right? So over here, sometimes the assertion can be true and the reason can be true, but there is no correlation between them. Right? The reason is not the correct reason for assertion. So sometimes, so if such statement arises, then you have to choose option B. Sometimes both of them are correct and the reason is the correct explanation for assertion. During that time, you have to select option number A. Right? So this is the example over here. Now, this question is based upon this particular diagram in your textbook. Right? This is the PBR322 vector, the plasmid, the genetically engineered plasmid. Now, in the first statement, in the assertion, it says E. coli, which is having this particular plasmid in its cell, if there is an insert of DNA, right, at BAMH1, so BAMH1 is right over here, right? BAMH1 is a restriction enzyme which makes a cut right over here, right? So a DNA is inserted right over here. So such types of bacteria won't be able to grow in a medium containing tetracycline. Okay, so that is correct. Now, why is it correct? Because the recognition site of this particular BAMH1 restriction enzyme is present in the tetracycline R region of the PBR322. So this particular gene, the pink region over here or the orange region over here, right? This one is the gene for tetracycline resistance. So the recognition site for BAMH1 restriction enzyme is located in tetracycline resistance gene. So that is also true. And now by applying our knowledge of insertional inactivation, we can say that the reason is the correct reason for this particular statement. The E. coli which contains this particular gene or this particular plasmid won't be able to grow in a medium containing tetracycline because the tetracycline gene has been inactivated. Right? This is insertional inactivation. Right? Why did it get inactivated? Because of BAMH1. It made a cut right over here and inserted a foreign gene over here. Because of that, the tetracycline gene got inactivated. So for this type of question, for this particular question, the answer is A. Both assertion and reason are true, and the reason is the correct explanation of the assertion. Is it clear? Right. So you have to select answer A. So you have to read the statements carefully. Okay, both statements. Let us look at another question of such type. So here, question number 11. Assertion says the primary transcripts in eukaryotic cells are non-functional. So you need to have a knowledge about the eukaryotic genes. Okay, and the transcription of eukaryotic genes. You have to know that. And there's a reason given over here, another statement, methyl guanosine triphosphate. It is attached to the 5' end of the heteronuclear RNA. Now, after reading these two statements, you have to make the selection like you did before, right? Now, over here, this question is based upon this particular diagram over here. We know that eukaryotic genes, right, or eukaryotic DNA or genes is a split gene, which means there is inclusion of both exons as well as introns. Introns are the non-coding genes, whereas exons are the coding genes. Okay, so exons, they code for proteins, whereas introns, they don't code for any proteins. So because of the presence of introns between exons, this particular transcript, the primary transcript, which has been transcribed from eukaryotic DNA, they are not functional. So the first statement, the assertion, is correct, right? So assertion is correct. So option D can be cancelled. Okay, so that is the thing, right? And second statement is the reason, right? Methyl guanosine triphosphate is attached to the 5' end of heteronuclear RNA. So if you look over here, methyl guanosine triphosphate, it is located on the 5' end of the transcript. So you can clearly see over here, right? This is the heteronuclear RNA, right? This is the heteronuclear RNA, and in the heteronuclear RNA, there is presence of methyl guanosine triphosphate at the 5' region. That is also true, right? So both the assertion as well as the reason, they are both correct. They are both true. Okay, so you can cancel out this C as well. Assertion is true, but the reason is false. You can cancel out this one. The reason is also true. Now you have to select between these two. Right? Now it says primary transcripts in eukaryotes are non-functional. Methyl guanosine triphosphate is attached to the 5' end of the heteronuclear RNA. Both the statements are true, but is there any correlation between these two? Does the reason support the assertion or not? Right? So we can clearly see that the reason does not support the assertion. The primary transcripts in eukaryotes are non-functional just because there are introns present between exons. Right? They are split genes. Introns are present between exons. So therefore, the primary transcript is non-functional just because of that. It's not because there is a G cap over here. Right? So therefore, the correct selection is both assertion and reason are true, but the reason is not the correct explanation of assertion. So the reason does not correctly explain the assertion. So you should select option number B. So in such type of questions, you have to read both of them, right, carefully, and then make a proper selection from here. Right? Don't jump to conclusions. Right? You have to apply all your knowledge that you have studied. Okay, so this is assertion reason type of question. Now the second type of question that is included in section A is, right, case-based questions. There are two case-based questions in section A. Right? In such type of questions, for example, question number 15, there is a paragraph given over here. There is a short paragraph given over here. You have to read the paragraph and then you have to answer 4 questions, which carries 1 marks each. Right? Now, there is an option over here. There are 5 questions given. You have to answer only 4. So that you can do on your own. Right? But the thing is, it is not checking your English. So it is not like an English reading passage. Right? So it checks your knowledge. For example, here, there is a paragraph given over here. It says ecological indicators. Right? And it talks all about dragonflies. right? How dragonflies, they are significant and positive ecological impact, right? How these dragonflies, they control mosquito population by eating mosquito larvae, both the nymph or the nymphal stage as well as the adult stage of the dragonflies, how they eat or they how they hunt mosquitoes, right? So that is given over here, right? And at the end there is one statement: community-wide mosquito programs that spray insecticides that kill adult mosquitoes, they also kill dragonflies. So here, right, you should apply the knowledge that you have regarding all the chapters in your syllabus, right? Like disease, human health and disease, and also in ecological chapters, right? You have studied about population interactions, right? So the knowledge gained from those chapters you have to apply over here, right? We will look at two questions depending upon or related to this particular question. So after reading this paragraph, you have to answer these questions, right? There are three more, but I've only shown only two, just to show that it's not English reading passage, right? So question number one: the approach to biological control includes. So here the main concept that they are checking is biological control, right? We have studied about biological control measures. How do we control a population of an organism by using another organism or its native enemy, right? So here, even though the passage does not say anything about biological control, it has just given an information regarding dragonflies. Here they have straight away asked question based upon biological control, so you should be able to answer here. Okay, and then two diseases less likely to occur in a region with plenty of dragonflies are. Then you have to apply your knowledge from chapter human health and disease, right? Which of all these diseases they are mosquito borne diseases, right? So you have to make a proper selection. Therefore there will be two questions, right? Question number 15 and 16, they are case based questions where you have to read a passage, a short paragraph, and then you have to answer four questions for each of them, which will carry 1 marks each. So there will be about 22 questions of 1 marks each that you have to attempt, right? So for some of you it will be very easy, it will be easier for you to score. But if you don't have proper knowledge, 1 mark questions are pretty tricky, right? If you don't know, you are just left, you have to just leave 1 marks. You cannot just write anything. They won't provide you 1 marks just because you wrote something over there. Okay, so you have to make correct selection. Okay, so that is the one. Now apart from that, all other questions, they are similar to your previous board examination questions, right? So therefore kindly go through the entire sample paper questions, right? So sample paper you have to download and go through the entire sample paper questions, the type of questions that they have given over there. And also the marking scheme is given. Marking scheme is nothing but the answer key for that particular question. So go through the entire sample paper question. You either download it, you print it out, just read through and look at the questions and look at the answers. If you can answer on your own, well and good. If you cannot answer on your own, you can refer to the marking scheme. But I advise you to try to answer from your own knowledge and then look at the marking schemes. Okay, so that is all.